Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the Pearson Edexcel International IGCSE Biology Paper 2B for January 2023. Let us begin with the first question. Question 1. Read the passage below. Use the information in the passage and your own knowledge to answer the questions that follow. The title is Treating Kidney Disease. Chronic kidney disease affects approximately 12% of the world's population, and many people are unable to obtain treatment. One method of treating chronic kidney disease is by dialysis. The diagram shows how kidney dialysis is done. So this is the patient's arm, and we can see we have two blood vessels. There is an artery and there is a vein. Blood is drawn from this blood vessel and is going to be pumped into the dialysis machine through the coiled dialysis tubing back into the other blood vessel, which is the vein. So down here they say, during dialysis, blood is taken from an artery in the arm and is pumped through a dialyzer. In the dialyzer, the blood passes through a long coiled dialysis tube made of a thin, partially permeable membrane. The tube is surrounded by dialysis fluid. The dialysis fluid contains glucose and ions at concentrations normally found in blood plasma, but does not contain urea. The urea passes from the blood into the dialysis fluid. The temperature in the dialyzer is kept at 40 degrees Celsius. After passing through the dialyzer, the blood is returned to a vein in the arm. Kidney dialysis can take up to 3 hours and must be done 3 times a week. Scientists have now designed a new bio-artificial kidney. This artificial kidney is a combination of engineering and living cells. The artificial kidney has a hemofilter made from artificial membranes that filter the blood. The substances filtered out of the blood then pass through a device called a bioreactor. This bioreactor absorbs useful substances back into the blood. The bioreactor is made of living nephron cells that are grown from stem cells. The cells are separated from the patient's blood by a silicone membrane to prevent the immune system rejecting them. Bioartificial kidneys are the same size as a human kidney and will be fitted inside the body to replace the kidney. The bioartificial kidney can react to changes in the body in the same way as a normal kidney. Moving on. Question A says, in 2021, the world's population was 7,900,000,000 people. Calculate the number of people in the world who have chronic kidney disease from line 1 and 2. If we go back to line 1 and 2, they said it's 12% of the world's population. So 12 divided by 100 times the population, which is given here, which is that, gave me this. But remember, they wanted us to write the answer in standard form. So to write this in standard form, it becomes that 9.48 times 10 to the power 8 is the number of people who have chronic kidney disease. In part B, urea is an excretory product that is released by the kidneys. Give the name of an excretory product that is released by the lungs. The lungs release carbon dioxide and water vapor, so any of these would be acceptable. Part C, explain 2 ways that the dialyzer is designed to increase the rate of removal of urea from the blood. Here, this is line 5 to 11. If we go back to line 5 to 11, they talk about the dialyzer being coiled. So the coiling is to increase the surface area. They also talk about the idea that it's partially permeable. This is to allow certain substances to go in and some not to go through. They talk about the idea that there is no urea, and this is to ensure that urea leaves the blood and goes into the dialysis fluid. They talk about the idea that it's kept at 40 degrees Celsius. This is to ensure that there is great enough kinetic energy for substances to diffuse at fast enough rate. So here I said the tube is cooled to increase the surface area for diffusion. Then a temperature of 40 degrees Celsius is used so that the particles have more kinetic energy for faster diffusion. And then the dialysis solution moves in a circulating manner to maintain the concentration gradient. This circulating manner is this manner I'm talking about, like that, and then circles in and out like that. That circulating manner is to maintain the concentration gradient. And then lastly, I say the dialysis solution does not contain urea to ensure a higher diffusion gradient of urea from the blood into the dialysis fluid. The hemofilter in the bioartificial kidney filters the blood. This is line 13 and 14. They say, where does ultrafiltration occur in the nephron? It occurs in the Bowman's capsule, so A should be our answer here. The cells in the bioreactor absorb glucose from the filtrate in a way that is similar to the cells in a nephron. This is line 14 to 17. Describe how the nephron absorbs glucose back into the blood from the filtrate. This reabsorption is selective reabsorption, and it occurs in the proximal convoluted tubule. Here, the absorption occurs against concentration gradient, where ATP is used in the process of active transport. And down here they say, the bioartificial kidney is connected to blood vessels and to the tube that transports urine to the bladder in the same way as the human kidney. Which row of the table is correct? The blood vessel bringing blood into the bioartificial kidney, that is going to be the renal artery. And the tube transporting urine to the bladder, this is going to be the ureter. So the answer is going to be A. Moving on. In part E, explain how the pituitary gland and the nephron cells in the bioreactor will respond in a patient who is dehydrated. Here we're talking about the pituitary gland as well as the cell in the bioreactor. So osmoreceptors in the hypothalamus will detect low water potential in the blood, and then the pituitary gland is going to be stimulated to release ADH hormone. This ADH hormone is going to stimulate the collecting ducts to be more permeable to water, so reabsorption of water into the blood can occur, and the produced urine is going to be more concentrated. So this brings us to the end of question 1. Let's continue to question 2. Question 2, the diagram shows the human eye. So here they've labeled part A, B, C, D, and X. This is the retina. This is going to be the iris. The pupil is that space there. This is the lens. This is the cornea, and then the conjunctiva. So they say which labeled structure is the cornea. Of course, that is going to be the cornea. And next they say, explain the changes in structure X as a person moves from a room where the light is dim to a room where the light is bright. Structure X is the iris, and it contains 2 types of muscles, circular muscles as well as radial muscles. So as you move from dim light to bright light, the pupil has to be constricted. So to do that, the circular muscles have to contract and the radial muscles have to relax so that the pupil size can be constricted. So I said the iris contains radial and circular muscles. Moving into bright light, the radial muscles relax and the circular muscles contract, leading to pupil constriction. Moving on. In part B, a student investigates if using one eye or using 2 eyes is better for judging the distance of objects. The student uses the plastic block shown in the diagram. The block has a grid where 6 different colored pins can be placed as shown in the diagram. So this is the block, and we can see the 6 different colored pins. This is the front, and the back would be that way. So they say this is the student's method. Place 1 pin into the lines of each column and draw so that each pin is a different distance from the front of the grid. Ask a volunteer to close one eye. Hold the grid in front of the volunteer so they can see the colored pins but not the grid. Ask the volunteer to call out the color of the pins in order of distance from nearest to farthest, and then record the number of pins that the volunteer identifies correctly. Repeat 8 different times, each time moving the pins to different positions in the grid. The student does the experiment again with both eyes open. Give the independent variable for the student's investigation. Here the independent variable can be the number of eyes. Remember, 1 time using 1 eye and then using 2 eyes. The next they say, give 1 variable that the student should control. The student should control the distance of the volunteer from the plastic block. They should control the light intensity, meaning it should be the same for all experiments, and then the size of the pins should be the same. Moving on. Table 1 shows the student's results. They say complete Table 2 by determining the mode and the median for the data shown in Table 1. Now remember, mode is the number that appears most, or the number with the highest frequency, and the median is going to be, of course, arranging the number in increasing order, and then the middle number is going to be the median. So if we look here, the number that is appearing most is a 3. We have 1, 2, 3 of them, and then we just have 2 2s, 2 0s, 2 1s, so there are more 3s here, meaning here our mode is going to be 3. You can see that's my answer here. And the mode here is going to be a 6 because we see 1, 2, 3, 4, 5, 6 6s, and the others are not as many, so that is going to be our mode. Now for the median, I arrange this in increasing order, beginning with the 2 0s, the 2 1s, the 2 2s, and the 3 3s, and then cancelled out. If you cancel here, you cancel here, cancel there, there and there, there and there. So we remain with the 2 in the middle. That is going to be our median. I did the same thing for this data set for using both eyes, beginning with the lowest number, which is 3, and then writing them in increasing order until the last number, which is 6. So cancel here with that with that, this with that with that, and 6 remains in the middle, so this is going to be our median as well. Moving on. The field of view is the area that an animal can see with their eyes. The diagram shows the field of view of a cat. This is seen by both eyes, seen by the right eye and the left eye. So they say explain why predators such as cats have 2 forward-facing eyes. Use the information in the diagram and the table to support your answer. Although there is this area that can be seen by both eyes, this side here can be seen by the right eye and this by the left eye, creating a wider area of space that can be seen by both eyes. So I said having 2 forward-facing eyes enables both eyes to see a wider area. Images to both eyes overlap, creating clear vision, so better judgment can be made in locating prey. Remember, these are predators. Their purpose is to find prey, so they need clear vision, meaning they need to also see a wider area so that they can make good judgment in order to hunt the prey. So this brings us to the end of question 2. Let's continue to question 3. Question 3. Hair color in cattle is controlled by one gene with 2 alleles. The allele for white hair, which is that, is codominant with the allele for red hair, which is that. They say heterozygous cattle are a pale red color called RO1. Question A says, state what is meant by the term gene. A gene is a section of DNA that codes for a specific protein. In part B, complete the table by giving the genotype of red cattle, white cattle, and RO1 cattle. Now, because these 2 alleles are codominant, it means there is no recessive in this nature. So red is going to have this genotype, white is going to have that genotype, and RO1 is going to have either that or that. Down here they say 2 RO1 colored cattle are mated. Determine the probability that the calf produced is a male calf with RO1 colored hair. Include a genetic diagram in your answer. Because we're mating 2 RO1 colored cattle, the parent genotype is going to be that for both parents, meaning the gametes are going to be either that or that. Now, by drawing this genetic diagram, we have that and that and that as possible gametes. So when fertilization occurs, the genotype of the offspring is going to be that, and that. So this is going to be the genotype, and the phenotype is going to be 1 red, 2 RO1, and 1 white. Now, to calculate our probability, they wanted probability of RO1, but also a male calf. So probability of male is 1/2. Probability of RO1 is going to be 2 out of 4, which is the same as a half. So a half times a half gives us 1/4, and therefore the probability is 1/4, or you could say 0.25. Some people can say even 25%, all those answers are acceptable. Moving on. In part C, in a group of cattle, there are cattle with white, red, and RO1 colored hair. Height of cattle is also a genetically controlled characteristic. In the same group, there is a greater variation in height compared with hair color. Explain why there is a greater variation in height of cattle than there is in hair color. If there is greater variation, then it's because height is a polygenic trait, meaning it is controlled by multiple genes. So those different variations that can occur in various genes are going to result in many variations of the phenotype. As I said, height is a polygenic trait. It is controlled by multiple genes that could have different combinations of recessive or dominant alleles for the different genes. Also, environmental factors like nutrition can affect height. Then lastly, describe how a farmer could use selective breeding to increase the height of cattle. So all they need to do is get specific cattle that are taller and mate them, then choose taller offsprings and mate them, then continuously mate taller offsprings for generations. So this brings us to the end of question 3. Let's continue to question 4. Question 4. Scientists have genetically modified soya plants so that the soybeans they produce contain less saturated fat. The scientists produced one transgenic soya plant. Micropropagation was then used to make multiple copies of the transgenic soya plant. Explain why micropropagation was used to produce copies of the transgenic soya plant. This is to ensure that we have larger numbers of plants in a shorter period of time that are genetically identical. So I say to produce genetically identical soya plants, this should be soya, in large numbers and at a faster or shorter period of time. there is no need for repeating genetic modification if we use micropropagation. Moving on. In part B, a student uses this equipment to compare the energy content of transgenic soybeans with the energy content of non-transgenic soybeans. We can see there is a boiling tube containing water, there is a thermometer, and then we have the browning soybean on a mounted needle. They say describe how the student could use the equipment to make a valid comparison of energy content of the two types of soybeans. Remember, when this is heated or burned, the heat release is supposed to be transferred to the water inside the boiling tube, and the thermometer is there to measure the initial and final temperature so that the temperature change can be calculated. So if we want to make a valid comparison, we need to use the same mass of the beans, and then we need to use the same volume of water. We need to ignite the soybean until it burns completely for all experiments, and then we need to measure the temperature rise, meaning initial temperature and the final temperature, and then calculate the temperature rise. Then we need to repeat the experiment under similar conditions in order to get a mean. Moving on. Here they say, give one safety precaution that the student should take when using the apparatus. In this case, they have to make sure their eyes are protected, and then they have to wear a lab coat. Then down here they say the diagram shows a different type of apparatus that can be used for the investigation. The sample is going to be here. There is a steel container. The thermometer is here. This is the water. And then there is going to be an ignition coil that is going to light the soybean. There is oxygen inlet, meaning oxygen is going to be supplied, so there is low chances of incomplete combustion. Then there is a stirrer to ensure even distribution of heat. There is an insulator to prevent heat loss to the surrounding. So this is going to be a more efficient method for heat transfer from the sample where it's heated to the water. So they say explain 2 reasons why this apparatus will give a more accurate energy value than the apparatus used by the student. Like I've already said, heat losses to the surrounding are minimized due to the insulator. Oxygen is supplied so complete combustion could occur or is going to be guaranteed. A stirrer ensures even distribution of heat. Then a larger volume of water used so less heat loss occurs. Then burning with an ignition coil eliminates the need to reheat. So this brings us to the end of question 4. Let's continue to question 5. Question 5. The graph shows the change in area of rainforest on the Earth from 1990 to 2015. So we can see area of the rainforest on the vertical axis in kilometers squared and the time in years on the horizontal axis. So they say determine the mean rate of decrease of the rainforest area in kilometers squared per year from 1990 to 2015. So here the decrease is going to be from 41200 to 40,000. So the decrease is going to be that minus that. We also need to find the change in time. This is going to be a period of 25 years. So the mean rate is going to be the change in the area of the rainforest divided by the change in time, giving me 48 kilometers squared per year as my answer. Moving on. Here they say loss of rainforest causes an increase in atmospheric carbon dioxide gas. Explain the negative effect of an increase in atmospheric carbon dioxide gas. If atmospheric CO2 increases, we remember CO2 is a greenhouse gas, so it's going to cause an increase in the global temperatures, or it's connected to global warming. So this causes the melting of the ice caps at glaciers, leading to flooding, and flooding can lead to loss of habitats. And climate change can also lead to loss of habitats, migration of species, extinction of species. This can also lead to increased landslides, soil erosion, ocean acidification, and so on. Moving on. In part B, scientists investigated the effect of deforestation on a river. The river flows through a forest, part of which has been deforested. They say the graphs show the changes in nitrate concentration and the biodiversity of animals in the river as it flows through the forest. On the vertical axis, we have nitrate concentration in arbitrary units, and on the horizontal axis, we have distance along the river. We see the nitrate concentration increases in the area of deforestation. Also, biodiversity decreases around that area of deforestation. So they say describe the role of microorganisms in the conversion of organic waste into nitrate ions in the river. When you're answering this question, you have to begin by stating which microorganisms are involved. So I said microorganisms like bacteria and fungi are decomposers. So in the decomposition, some nitrifying bacteria are going to break down ammonia into nitrites, and then these nitrites are going to be broken down by nitrifying bacteria into nitrates. Moving on. Here they say the number of different animal species living in the river was recorded as a measure of the biodiversity of animals. Give a reason why this may not be a complete measure of biodiversity. It may not be a complete measure because some species are going to be seasonal. They'll be there at a specific period of time of the year. Also, biodiversity should consider unicellular organisms. Remember here they said animals. They did not say organisms. So unicellular organisms like bacteria and fungi should be considered, not only animals. Next they say, explain the change in the biodiversity of animals along the river. They want you to use information from the graphs in your answer. I want to go back here. We can see from the area where deforestation has occurred, the nitrate concentration is increasing. And the increase in nitrate concentration correlates with a decrease in biodiversity of animals. This could be due to eutrophication within the river. So I said, as nitrate concentration increases, biodiversity decreases. Due to deforestation, nitrates are washed into the river or leached to the river. So eutrophication takes place due to the increased algal growth. Remember, these nitrates are going to be like boosters for algal growth. So there'll be less light entering the river, there'll be less photosynthesis that occurs, and the dead algae are going to be decomposed by the microorganisms. This process uses up dissolved oxygen, so the aquatic animals will die due to lack of oxygen, and the biodiversity is going to decrease. So this brings us to the end of question 5. Let's continue to question 6. Question 6. Rabies is a dangerous disease caused by a virus. The rabies virus is a pathogen. Which other groups of microorganisms include pathogens? Bacteria and fungi are pathogens, but also some protists are pathogens, so the answer here should be a B. And in part B, rabies is transmitted to humans when they are bitten by an infected animal. Domestic dogs are dogs kept by people as pets or as working animals. Dogs and some wild animals are known to transfer rabies to humans. In the 1940s, a rabies vaccine for animals was introduced in the United States, and most domestic dogs were vaccinated. The graph shows the number of cases of human rabies and domestic dog rabies in the United States from 1938 to 2018. On the value axis, we have the number of cases for humans as well as for domestic animals. On the horizontal axis, we have the time in years. After introduction of the vaccination in 1940s, we see the number of cases decreased drastically and remained oscillating about a specific value. So moving on. Here they say the immune system of dogs works in a similar way to the immune system of humans. Explain how the rabies vaccine prevents dogs from developing rabies. This is because the vaccine has specific antigens, and these antigens are going to trigger the immune system in order to produce memory cells as well as antibodies. So on encountering the real rabies virus, secondary immune response is going to occur, memory cells will be activated, and antibodies are going to be produced within a shorter period of time and in large numbers. So I said a vaccine uses a weaker or inactive virus. The antigens trigger immune response, antibodies are produced by lymphocytes, and then memory cells are going to be formed. If they encounter the rabies virus, secondary immune response occurs, memory cells are going to be activated to produce antibodies faster and in large numbers. Down here they say, discuss the effects of the use of the rabies vaccine to immunize domestic dogs on the number of cases of rabies in humans. Use the information from the graph to support your answer. To be more specific, I'm going to change this to 1940. Before 1940, the rabies cases were high. After introduction of the vaccine, the cases fell. However, a correlation exists between the rabies cases in dogs and in humans. The rabies cases are not completely eliminated, but fluctuate about a value below 2,000. Also, overall, there have been more cases in dogs than in humans. Also, the data has been collected for a long period of time, so it can be reliable. Moving on. Here they say a new RNA vaccine is being developed to treat rabies. RNA that codes for parts of the rabies virus protein code are injected into the body. Cells then take up the RNA and produce the viral protein. Describe how the cells use the RNA to produce the viral protein. This is about protein synthesis. So we know RNA that is going to be taken up by the cell is going to bind onto the ribosomes for translation to occur. Then the transfer RNA with an anticodon complementary to the RNA codon will deliver a specific amino acid, and these amino acids are going to be joined together in condensation reactions to form a polypeptide. So this brings us to the end of question 6, as well as to the end of this whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.